So today we're going to be talking a little bit more about how we can visually and intellectually understand what's going on with the dominant and the recessive alleles. Talk a bit more about the phenotype, the genotype, the heterozygousness, and the homozygousness of alleles and chromosomes and all of that. But before we do that, I just want to quickly go over some of those definitions from the last video. So again, something that's a diploid, it's going to have two sets of chromosomes. Most organisms are diploids. When we get to the gametes, we've got half the genetic information, like we talked about last time, making it haploid. An allele is a possible expression of chromosomes or genes. So an allele could determine, how tall you are, what your general build is, if your second toe is longer than your big toe, if your earlobes are connected to your head, or if there's like that little gap in between your earlobe and your the side of your head, your jaw, I don't know, what color your eyes are, how long your eyelashes are, what color your hair is. All of those expressions are alleles. Okay, so the gamete, those are the sex cells, the egg or the sperm that contain half the genetic information. The gametes come from the parents, and then they join together, and then that becomes the new organism. Okay, we have dominant alleles, and that's the stronger characteristic that masks the weaker one, even if only one dominant allele is present. We have recessive alleles, and that's a weaker characteristic, and you have to have two of the recessive ones to make sure that they're not going to be masked by a dominant allele. Phenotype is how something looks, how the chromosome or the alleles are expressed visually. So what color hair do they have? What color eyes do they have? How tall are they? Those kinds of things. The genes are going to be talking about, do they have both dominant? Do they have one dominant, one recessive? Those kinds of things. That's the genotype. So within the genotype, we can have the heterozygous, where they have different alleles and different chromosomes, or they can be homozygous, where everything can be the same. And if they're heterozygous, it's going to look, we're going to see the dominant characteristic, the dominant allele. And if they're homozygous, we're going to see, whatever that is. Okay? So let's look at the way that most people figure this out is traditionally with this table or chart, and it's called a Punnett square, if you've heard about those before. So this is one Punnett square, specifically working with peas, because that's what Gregor Mendel used. And over here we see we have a pure breeding round yellow pea seed. Okay, and we can see here we've got RRYY. Now those are what we're using to represent the alleles. So since it's pure, that's another word for homozygous. That's supposed to be an H. Sorry. Zygous. Okay. So homozygous, so we have two R's, which is for round, and two Y's, which is for yellow. Now traditionally, dominant letters, and these letters really can be whatever you want, but traditionally, if we're looking for a particular characteristic, we pick the first letter of that characteristic. So in the case of peas, being round and being yellow, those are dominants, so we pick round, yellow, they're dominant, they're capitalized. Now down here we have, so this is homozygous dominant. And now down here we have a pure breeding wrinkled green seed, and you can see they're lowercase. That means it's recessive. So we've got homozygous. I'm going to write on top of our letters, oh well. Recessive. You can see we're not changing our letters, but we are making them lowercase to show that they're different. So now they're going to breed. We're going to cut those chromosomes in half, make it a haploid cell. We get one set of dominant round, one set of dominant yellow, one set of recessive wrinkled, one set of recessive green, and that's going to give us a heterozygous, running out of space, a heterozygous P. Now you can see it looks like the pure dominant, but you can see from that here it looks like that, so that's the phenotype, but here is the genotype. We've got dominant recessive, dominant recessive. All right, and oops, sorry. Now we're going to put that information into this lovely chart. Okay, this is the Punnett square. So we've got all the different possibilities here. Now this one is a bit complicated because we're looking at two characteristics at the same time. So we're going to take two of these heterozygous P's and breed them together. So one possibility for one of the gametes would be dominant round, dominant yellow. Okay, or we could have recessive round, dominant yellow, recessive round, or I'm sorry, dominant round, recessive yellow, and then recessive round, recessive yellow. And again, we can have those on the top as well. And so now we're just going to be putting them together. We're going to be combining them because, you take one gamete from one parent and the other gamete from the other parent. They're haploid cells. You put them together, you get the diploid cells, and then you get your full chromosomes that you need for the new organism. So we've got dominant plus dominant, two dominants, dominant, dominant, two dominants, round, yellow. Okay, recessive dominant. Dominants are always written first, and then dominant, 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 dominant. Still, even though here we have heterozygous, the dominant is going to show, so it's still going to look round. Again, dominant, dominant. And then for the yellow, we've got dominant and recessive, but again, that dominant is going to show. Dominant, recessive, and the dominants are going to show. Next round, very similar, but now, since this one has a recessive and this one has a recessive, we start to get those recessives, those homozygous recessives, and they're showing. So here, their dominant's covering up. There's homozygous. We've got capital Y, so everything in this. So those are going to be dominant. This here, we still have a dominant Y, and so it's going to be dominant. Yeah, since this is a dominant, everything in here is going to show dominant. But now again, we've got recessive and recessive, so that shows the recessive trait. But again, since we have that dominant from this parent, it's still going to show as the dominant. All right. So now, kind of clean this up a little bit. Since we have now dominant here, everything is going to be round. But now we have a recessive in the yellow, so now some things are going to start being green. So we've got dominant, dominant gives us two dominants. Recessive, dominant, dominant, recessive. Dominant, recessive, dominant, recessive. Recessive, dominant, dominant, recessive. Dominant, dominant, nice and round, but now we've got recessive and recessive. Both of those are recessive, and we get the green P. Dominant, recessive, we get the round. Recessive, recessive, we get the green. Now over here, again, the same thing, but now we've got recessive for the round, and so we're again getting those wrinkly ones. We've got recessive for the color, so we're getting some green. Okay, so that's generally how it works. We'll look at another one that doesn't have quite so many characteristics and will be a little bit easier to follow. So now we've got two generations. Okay, so first generation, we have a pure red sweet pea flower and a pure white sweet pea flower. So dominant over here, recessive over here. They're all going to combine to heterozygous heterozygous genotypes, but still dominant phenotypes. They're all going to look red, but they have both the dominant and the recessive. So now these over here in our second generation, these are going to move to our second generation, and they've got dominant and recessive. So we've got dominant, recessive, dominant, recessive. So here in this one, both the dominants come together and we get another pure red rose, because now it's back to homozygous dominant because we've got both of those dominant characteristics showing. In this one over here, we've got dominant and recessive, so it's a hybrid. We've got over here dominant and recessive, we've got a hybrid. And now down here, we have both of those recessives, and we get a pure or homozygous white rose. Okay, so let's try another one on our own. Okay, so something that I believe in humans is determined by just one allele is such a lovely topic, but like how wet or dry your earwax is. Okay, and I'm not sure exactly how it goes, so we'll just say that wet earwax is dominant. So somebody is going to have wet earwax, heterozygous, and then somebody else will go ahead and say they're going to have recessive, which would then mean dry earwax. So let's say this is mom and this is dad, not that it matters. So now we're going to break this up. So mom is going to have the capital W and the lowercase w, and dad has lowercase, lowercase. So we're going to bring this recessive for mom down to both of these kids, assuming that there's 4. But, you know, there would be 4 between the two. There would be the 4 different possibilities. Okay, and now we bring this recessive from dad across, and this recessive from dad across, and this one comes down. Writing the Punnett squares isn't terribly difficult. You then have to figure out how to read them. So here we've got a capital W, so this is going to have, let's say 1, 2, 3, 4. So 1 and 2 are both, you can see, they both have that capital W. So the phenotype is going to be dominant. They're going to have wet earwax. But the genotype, they're different, so we have heterozygous. I'm just going to write hetero because it's a little bit shorter. Now on 3 and 4, we've got the same. So the phenotype is going to be recessive, and the genotype is going to be homozygous. Again, of course, recessive. Ugh. Can't write homozygous for whatever reason today. Okay, so let's try that again. Okay, so now we're going to do one that's facetious, and we're not going to use the right amount of chromosomes, because in reality it takes, you know, more than one allele, more than one chromosome to determine your hair color. But let's say we have a mom who has brown hair, and she has just homozygous brown hair. And then we have a dad who has brown hair, but he has heterozygous brown hair. Okay? So we draw our nice squares, and we split up. That's a very big center square. Oh well. We split up our attributes, so moms go on top. It doesn't actually matter. Dads could go on top. It's fine. And now we just fill in our chart, bringing these down and these across. So B, B, B. I'm working down first, and now I'm going to work sideways. Okay, so we've got 1, 2, 3, 4, where in this case, 1, 2, okay, so our phenotype, 1, 2, 3, and 4 are all going to have brown hair. They're all going to be dominant because we have these capital B's everywhere. Okay, so they're all going to look like they have brown hair. But for our genotype, okay, so 1 and 3, they're going to be homozygous and homozygous dominant. And 2 and 4, okay, so that was 1 and 3. 2 and 4 are going to be heterozygous dominant. Okay, so now that matters because let's say person 2 grows up, has a kid with somebody who is, let's say, they're blonde or redhead or whatever, but they don't have brown hair. As again, it takes more than one gene, but that's okay. So we're going to take their dominant brown hair gene sideways, this recessive one, or I'm sorry, dominant brown hair gene down, recessive down, recessive sideways, sideways. And now over here, look, even though this person, dad, whoever, looked like they had brown hair, over here they've got 2 kids that don't have brown hair. They've got blue hair, or they've got blonde hair or red hair or whatever. Okay? So, you know, it can change the way it works. Now, let's see. That was the same. Okay, so I did that one kind of again. So let's say we have one person who has, I don't want to use B's anymore. We'll do eye color and we'll just use other letters. Okay, so let's say we've got 2 people with brown eyes. Brown again is dominant, but they're both heterozygous dominant. So that means they've got one dominant allele and one recessive allele. Now again, in a perfect statistical genetic world, they would have 4 kids. And these alleles are going to go down. These are going to go down. And these are going to go across. And these are going to go across. And now, with 2 heterozygous, we've got 1, 2, 3 that are showing phenotypes of dominant eye color. So we've got 3 kids with brown eyes. But now we have over here this one person who's going to have recessive blue eyes or recessive green eyes. And then we've got 2 genotypes that are homozygous, one that's homodominant, and one that's homozygous recessive. And then, I don't know, 2 that are heterozygous. Okay, so hopefully that gives you enough of an explanation to get a start on those genetics problems, on the Punnett square problems. We will definitely be talking about this during the live chat. We'll be going over a few more examples. If you have any questions about those, go ahead and email me. But hopefully this will keep you going until we get to the live chat. All right, talk to you later.